disease. From the name itself, you can guess what is happening. If there will be a cut in the body, usually within 10 to 14 seconds, the blood is clotted. So that we are at the safe side. But there are certain persons, due to presence of some defective genes, if there is a slight cut in the body, continuously it bleeds on, and sometimes it may lead to death. This is known as hemophilia or bleeder's disease. So this is the symptom, that means bleeding will not stop. But what is the cause of this disease? If we discuss the cause, usually the cause of this disease, it is usually a sex-linked recessive gene. Now usually this gene is present in the X chromosome. And what is the function of this gene? Usually this gene produces certain proteins which helps in the clotting of the blood. So what usually happens? Usually in the X chromosome there is one gene, we can call it as F8. Usually it produces a factor that is 8. So this factor 8, if this cannot be synthesized due to defect in this gene, then it will cause hemophilia A. Similarly, suppose the gene F9 is there. This is responsible for factor 9, and if it is defective, then it will give rise to hemophilia B, and this is also known as Christmas disease. Christmas disease after the name of the patient known as Christmas. First it was reported in a 5-year boy, Christmas, so accordingly it is named as the Christmas disease. So usually what is the main function of these factors? For blood clotting, the protein is fibrin. But how this fibrin is usually the fibrinogen is converted to fibrin, and this fibrin is the main framework protein, because it is the fibrous protein, and this is responsible for the clotting of the blood. But for the conversion of this fibrinogen to fibrin, it requires thrombin. So where from this thrombin will come? Now usually this thrombin will be synthesized from prothrombin. So when prothrombin is converted to thrombin, then it is the function of the thrombin which will convert the fibrinogen to fibrin, and this fibrin is a fibrous protein which is the main framework to clot the blood. And this prothrombin, it converts this to thrombin only in presence of these factors which are synthesized by their respective genes, and genes are present in the X chromosome. So if these genes become defective, then in this case it will lead to hemophilia A, in this case it will lead to hemophilia B, and in that case the bleeding will not stop. So this is the main cause. So this is the important, it is a sex-linked recessive gene. Now how this gene is inherited, let us discuss that part. Inheritance of the gene. So usually this gene is present in X chromosome, and you know that in case of the human male, there is one X chromosome and one Y chromosome. Now the defective gene, suppose the defective gene is H. Suppose. Now, if this defective gene will be present in male, then it will be X, H, the H is superscript and Y. That means this gene has no other allele in Y. That means it will express itself and the person will be hemophilic. That means there will be bleeding. But in case of females, what happens? In case of females, there are two X chromosomes. So if one X chromosome will have that gene, then it will not be expressed, because in the other X chromosome, the dominant gene which is present, that will suppress itself, so it will not express itself. So in that case, the female will be only a carrier. But by chance, due to some reasons that we'll discuss, if both the X chromosomes will have this one, then what will happen? If both of them are there, then it will be hemophilic. It will be hemophilic. But in this case, it is completely lethal, and that person will die. So, in case of males, since it is present in X chromosome, it is usually expressed. But in case of females, if it is present in one X chromosome, it is not expressed. Only if it will double, which is again rare, then only it is hemophilic and lethal. That is why in males, the hemophilic condition is more prominent and more common, while the females act as carriers. This is the method of inheritance. Now let us give some examples so that our understanding will be better. Now let us take one example, first example, where a normal female means double X, and this normal female will marry one hemophilic husband. So naturally hemophilic husband will be XHY. Now here the female will produce two types of gametes, and the male will also produce two types of gametes. Now if we draw the Punnett square, and we will take the female in this side and male in this side, then it will be X, and this will be X, H and Y. Now you see, this will be this one will be X and XH. This will be X and XH. While in this case it will be XY, and in this case it will be XY. Now you see, where double X are there, they are daughters. So in daughters only one X chromosome has that gene. That means both the daughters. That means two daughters are carriers. And what about the sons? The two sons are normal. That means two daughters are carriers. That means 100% daughters are carriers. Or we can tell that 50% of the children or all the daughters, they are carriers, while all the sons are normal. Now let us take the second example. Example number 2. So in example number 2, let us take a carrier woman. So naturally carrier woman will be X and XX. And if this carrier woman will marry to a normal man, so normal man will be X and Y. So it will produce two types of X, and the male will produce two types of sperms. Now in the F2 generation, what is happening, you see. In the F2 generation, it will be X and XH because it contains that gene. And in this side, this will be X and Y. This is the male side and this is the female side. Now you see, this will be X, X. This will be X, 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 H. This will be X, Y. And this will be X, H, Y. Now you see what is happening. There are two daughters and two sons. But what about this daughter? That means one daughter is normal. This one. And one daughter is here. Here one daughter is carrier. Because this is carrier. While this son, one son is normal. And what about this son? This son is hemophilic. That means one son is hemophilic. That means out of two daughters, one is normal and one is carrier. That means 50% of daughters are normal, 50% daughters are carrier. And among the sons, 50% sons are normal, while 50% sons are hemophilic. Now let us take a third example. Only by this method you can solve any problem that is asked in the examination. Suppose example number 3. Let us take a carrier woman. Carrier woman. So carrier woman means XH. And suppose this carrier woman will marry one hemophilic husband. Hemophilic husband. So hemophilic husband means XHY. Now you see, in the female there will be production of two types of eggs, and in this side also there will be production of two types of sperms. Now let us draw the checkerboard. Now in the female side it will be X, XH, and in the male side it will be XH and Y. This is male side, this is female side. Now you see, this one will be X, XH. This one will be XH, and from this side is also XH. Now come to this, it will be X, Y, and this one, it will be X, H and Y. So these two are daughters, these two are sons. Now what is happening you see. What about this daughter? This is a carrier. That means one daughter is a carrier. What about this daughter? Then one daughter is a hemophilic. So when one daughter is hemophilic, this daughter will die because it is lethal. Then what about this son? This son is normal. One son normal. And what about this son? This son is hemophilic. That means depending upon the character that is present in case of the man and the woman, what type of offspring should be produced, that can be easily obtained by this method. So we have discussed 3 different types of examples. Now one important thing is where this hemophilic condition is found. This is also known as royal disease because it was first reported in the Queen Victoria, the Queen of England. Actually, when it was detected that it's present in Queen Victoria, it was found that none of the Victoria's ancestors, they have this character. Then how it came to existence? So there must be some mutation in her father's genes or in mother's genes or there is a third probability that there may be some mutation in her own sex cells. But anyway, Queen Victoria gave birth to a son, and the name of son is Leopold. Leopold. So naturally, the Leopold was also hemophilic. Hemophilic, and also Leopold fathered to a daughter. That means he has also one daughter. So naturally the daughter was a carrier. Now in addition to Leopold, Victoria had also two daughters, and those two daughters they married in the royal family of Spain and Russia. So at present this hemophilic disease or bleeders disease, it is also found in Russian royal family, in Spain, and also in England. And if you ask me how it can be cured, it is very difficult to cure because it is due to defect in gene. But sometimes there is a reverse mutation, back mutation, and if that happens, then it can be cured. So after this, let us discuss questions, because the questions are very important in this case. So let us discuss two MCQs. So one MCQ, let us discuss the daughter born to a hemophilic father and normal mother, and a normal mother could be first choice normal, second choice carrier, third choice hemophilic, and fourth choice all of the above. So this question was once asked in AIMS, and by drawing the checkerboard easily you can solve it. Now let us discuss the second question. A man known to be a victim of hemophilia married a normal woman whose father was known to be a bleeder. Thus, it is expected that first choice all of their children will be bleeders. Second choice, half of their children will be bleeders. Third choice, one-fourth of their children will be bleeders. And fourth choice, none of their children will be bleeders. And again, this question was also asked in AIMS. And only by drawing the checkerboard, for drawing the checkerboard you will take only hardly 10 seconds, but your answer will be perfect. So thank you very much for watching the video. I hope you have a clear understanding about the bladder disease or hemophilia. So thank you very much. God bless you. Have a good day.